What we'd like to do for the next hour, again, is a little different from the standard presentation. I'm going to talk for about 20, 25 minutes, talk a little bit about emerging pathogens, just, kind of what they are, where do they come from, and also talk about the work that we are doing in Haiti, specifically related to cholera. And then we're going to switch over and expand the size of the group and broaden the topic range with a particular focus on some of the viral pathogens that are making headlines right at the moment, including the MERS-CoV, which is creating a little bit of concern in Saudi Arabia. So this has been called the age of pandemics, probably a slight overstatement, but nonetheless, you've heard for the past hour about the symbiotic relationship between lice and humans. Humans also have pathogens that sort of co-evolve with them, some with greater success than others. Pathogens, of course, are a little different relationship because they do bad things to us. And so consequently, I think the thing to emphasize is that this is also a very dynamic evolutionary process. The thing about emerging pathogens, to me, that makes the field exciting is that there is constant change. You never know what's going to show up literally tomorrow. Bacteria, viruses evolve very rapidly. They constantly are filling in new niches, coming in, doing new things, taking advantage of new opportunities. And so, unlike some of the more classic diseases chronic diseases, where there's sort of one disease, you understand it really doesn't change that much. When you're talking about infectious diseases and particularly emerging pathogens, it is a field that is in constant change because the microorganisms that you're dealing with are constantly evolving. So briefly, a slight ad here at the beginning. We are at the Emerging Pathogens Institute at the University of Florida. EPI, or Emerging Pathogens Institute, is an interdisciplinary research institute. It was initially created with a $60 million appropriation from the Florida State Legislature. And again, we don't just do humans. We focus on animals, plants, and humans. And again, this gets back to a concept of what has been termed One Health. The idea that when you're talking about pathogens, you really can't be parochial and just focus on humans because it's a dynamic process that cuts across, particularly between animals and humans. We've got over 200 faculty members from 10 University of Florida colleges who are affiliated with our institute. This includes faculty from colleges of medicine, public health, veterinary medicine, agriculture, all the way to the College of Law. Again, we also have a strong global emphasis. Pathogens do not recognize national borders. They don't like showing visas to come across. And so consequently, if you're truly going to understand emerging pathogens, where they're coming from and what the risks are, you've got to do it at a global level. So what's an emerging pathogen? Technically, a disease-causing microorganism that is newly recognized and or increasing in frequency in human, animal, or plant populations. Sometimes the easiest thing is just to give some examples. And again, you can usually look at the headlines and get a feel for what your new favorite emerging pathogen is. H1N1, swine flu, pandemic flu, and actually we'll talk today some about the H3N2. The Middle East Respiratory Syndrome, or MERS-CoV. AIDS is an older player, but clearly has been a major emerging pathogen over the last several decades. West Nile, dengue, chikungunya. On the plant side, for us here in Florida, citrus greening has been a major emerging pathogen that has devastated over a third of the Florida citrus industry. Multi-drug resistant TB, drug resistance in human pathogens. Again, CDC just put out a new white paper that described the major problem we're encountering in this country with the emergence of antibiotic resistance. And then things like E. coli 015787, foodborne pathogens, and the list could go on and on. So why do pathogens emerge? There are basically three categories. You can get the emergence of a genetically new strain. So it's truly something new. The genetics have changed. This is something new and different that has evolved and is now starting to threaten humans. Secondly, there can be changes in opportunities for an established pathogen or a known pathogen to spread, to grow and spread within our world. And these are usually what we call anthropogenic. We do it to ourselves because of ways in which we change our environment, the ways in which we change the way we process foods. These types of things will suddenly open up a niche where a pathogen that's been out there all along suddenly can go, hey, wow, I can start moving in here and doing my thing. And so while we talk about emerging pathogens and the genetically new pathogens, in actuality, probably a much greater problem is the spread of older pathogens, which either we thought were under control or we really didn't know about, into new niches, which we have opened up because of things that we're doing to our world. And finally, changes in host susceptibility. This is starting to be an issue. As you know, as all of us get older, we become more susceptible to infections. And so consequently, because of changes in general population susceptibility, we begin to see the spread of new diseases or diseases that previously had not been able to spread with a similar ability. Now, one of the things I would emphasize is that when you talk about, where do these things come from, probably about 70% of newly emergent pathogens come from animals. They're what we call zoonoses. And so there is this easy pathway that goes between animals and humans. And that tends to be the source for a lot of these pathogens, both those that have newly evolved and also those that have been there all along, but because of our increasing contact with animals or because of some changes in the relationship there, suddenly find that, yeah, humans are not a bad population to move into. And finally, pandemics. I started out with that slide saying the age of pandemics. Essentially, a pandemic, by definition, an explosive, frequently reoccurring disease that spreads rapidly through populations and across continents. So here we're talking about something that doesn't just spread around your neighborhood, but something that truly has the ability to move rapidly at a global level. Why do pandemics occur? Frequently, you're dealing with pathogens that have an environmental or animal reservoir. So they're kind of sitting there waiting. And then frequently there is a genetic structure that encourages reassortment, recombination, which results in the appearance of new subtypes, which again can move rapidly. Generally, you need to have easy person-to-person transmission in a sustained way. If something's going to spread globally, it's got to be able to jump from person to person pretty quickly. And to catch everybody's attention, usually we're talking about things that are able to cause serious illness. And we're going to talk today about some of these potentially pandemic or pandemic emerging pathogens that are of concern today. So I'm going to start by talking a little bit about cholera. Now, cholera is a very ancient disease. Cholera has been present in association with humans for millennia. If you go back to the ancient Indian literature, it is clear that it has been around for thousands of years. It causes such a distinct clinical syndrome that you really can't miss it when it occurs. And it has a reservoir. It appears to have evolved from and to maintain a major reservoir in the Bay of Bengal. And this is a boat out on the Bay of Bengal. This is the home of cholera. It's a disease that has attracted a great deal of attention, including a lot of literary attention. This is actually one of my favorite poems from Kipling from 1912. We've got the cholera in camp. It's worse than 40 fights. We're dying in the wilderness, the same as Israelites. It's before us and behind us, and we cannot get away. And the doctors just reported we've 10 more today. And I think for those of us who work in cholera, there is an understanding of sort of the sense of desperation. All of a sudden, it's everywhere. You've got cases coming in. What are you going to do with them? And you see the spread, and it doesn't stop, and it keeps happening. And so, at least in Kipling's time, this was a devastating disease. Case fatality rates of 40%. It kills people, and it moves rapidly through populations. And the thing is, it's still a bad disease today. We think about a lot of the infectious diseases as going away. Cholera is not going away. We actually have more cholera cases today than we've had in the past. And this just shows numbers of cases reported. numbers of different countries. We are seeing truly cholera at a global level. Where does cholera come from? Well, again, just to get a little bit into the science, as we talk about transmission, we start with an environmental reservoir. Vibrio cholera is very happy out in the environment, particularly estuarine areas with a little bit of salinity or fresh water in rivers, ponds. It lives there, and it can live there literally forever. It's happy in the environment. However, under certain environmental conditions, certain changes in temperature, and we find that temperature is probably the major driver, then cholera undergoes what we call a bloom. The Vibrio cholera in the environment begin to really start multiplying. And then what you get is spillover into human populations. People go use the pond water or the river water for drinking and bathing and washing their dishes. And if there's a lot of Vibrio cholera in that water, then some human's going to get it. But once it gets into human populations, then it takes off. It rapidly accelerates. It moves in genetically. It shifts gears. It turns off the whole set of genes it used in the environment, turns on a whole new set of genes that it uses in humans. And suddenly, as it passes through humans, it becomes hyper-infectious. It has the ability to very rapidly move from person to person, and suddenly you've got epidemics and pandemics. And this is what you get. This is the cholera ward in the cholera hospital in Dhaka, Bangladesh. In many ways, cholera is a simple disease. Basically, what cholera does is cause really severe, profuse diarrhea. You can have as much as 1 liter of diarrhea per hour. Given that your entire blood volume is about 5 liters, it doesn't take very long to become severely dehydrated, and you can die within 12 to 24 hours with circulatory collapse. However, if you put the fluid back in, then you do just fine. And so what you see is intravenous lines going into these women. And again, for those of you who are not familiar with cholera treatment, these women are laying on cholera cots. Basically, it's like a camp cot, rubber cot. It's got a hole strategically cut in the middle. And the patient is, again, appropriately positioned over that hole because they're passing vast amounts of diarrhea just effortlessly. You see that bucket down underneath the cot? Basically, the nurse goes around about every 15 minutes, sticks a ruler in the bucket, finds out how much diarrhea they're having, and then the goal is to replace the fluid going in by 1.5 times the amount of diarrhea that's come out. And if you do that, everybody lives. Fatality rate for cholera is much less than 1% if you put back in the fluid. However, the key to it is oral rehydration. Now, for millennia, we have dealt with this disease, and people have died. But it took some very sophisticated science in the 1960s, which first discovered that you could bypass the absorption tracts that are blocked in cholera by adding glucose to your salt solution. And this is, again, it's a fairly complex story. The bottom line is, if you give somebody a sugar-salt solution, then they can absorb the water. And so consequently, that has led to the development of oral rehydration, which I think is probably the major medical advance in the last century. It has probably saved more lives than any single thing we've done. We've got all our fancy drugs and CT scanners and all of that. This is what saves lives at a global level. However, we still have cholera. This is countries reporting cholera cases in 2011, as well as case fatality rates. Remember, I told you the case fatality rate should be less than 1% when you're dealing with somebody who knows what they're doing. Now, the darker colors are the countries that have case fatality rates over 1%. And essentially, you can see that's sub-Saharan Africa. That's where we don't have the medical care to be able to provide the support for these people. So let's skip to Haiti. January 12th, 2010 was the earthquake. It devastated the island. And at the same time, there was almost total destruction of the public health infrastructure, including water and sewerage. It was a setup for epidemic disease. And everybody said that. And I and others who are specialists in cholera always added, but there hasn't been cholera in Haiti for 100 years, so it's probably not going to be cholera. But then, all of a sudden, actually October 21st was the first diagnoses coming out of the labs at the National Public Health Lab. First cases were found along the Arbutinite River, association of cases with the river, and what was very interesting was that there was a UN unit from Nepal at the epicenter of the outbreak. Their camp, the sewage from their camp was not being treated as it was dumped into the river. The treatment facilities had broken down, and so they were essentially dumping raw sewage into the river. And again, what was found was that the isolates coming, at least at the beginning of the epidemic, were all clonal. They were all identical. And for those of us who do this type of work, when you find clonal isolates, it suggests that you have a single common source. So again, while this remains highly controversial, and as many of you are aware, there are international lawsuits going on with relation to this, I think the evidence is extremely strong that what we had was probably an asymptomatic carrier from Nepal who was part of the Nepalese peacekeeping groups who defecated into the sewage in the camp and it got into the river, and we suddenly had an epidemic. Now, University of Florida has an infectious disease field lab in Haiti. We've been involved in research there for a number of years. This is actually the lab, somewhat, it's not that impressive, but it actually has the capabilities of a full laboratory here in the United States. Jimmy Carter was there for the opening of the lab. It's a nice setup. And we, too, were involved in some of the early molecular genetic studies. Our studies confirmed what other groups were finding, that indeed these were clonal isolates, almost certainly from a single source. And then what happened was you got very rapid spread across the entire country. In terms of science, one of the things we did was work with the development of mathematical models to try to predict where the disease was going to go, how it would spread. Again, these are calculations of what we call R sub naught, which gives a sense of the force of infection. Again, the initial intensity was around where the first cases were, and then the intensity decreased as it moved through the rest of the country. Again, these are the types of data that we're able to get using mapping and the other tools that we have in hand, modeling that allow us to sort of predict and understand and prepare for epidemics. In the area where we work, we actually use GPS to identify all of the case households. And again, what we found was that as you move particularly through this area, we could actually literally track the spread of the disease along roads, along the major highway. You could watch it on a day-by-day basis as it spread along the highway. And then it would move out into the countryside, and then it would seem to get into the rivers, and then you could track it spreading down the rivers. So again, by using GPS, modeling, and a variety of other techniques, it let us actually monitor, literally on a day-by-day basis, the spread of this particular pathogen. So the question comes, are we establishing an environmental reservoir in Haiti? Because if we do, then we're going to have a disease that's going to be around for quite a while. Remember I said it can live in the environment for decades, if not forever. And actually, we've been doing studies. We've been regularly monitoring a series of rivers and other water points in the area where we're doing most of our studies. And unfortunately, what we find is that the bacteria does appear to be taking up residence within the environment. Not totally unexpected, but very concerning, because it means that what we're seeing is not just sort of a one-shot thing, but something that's going to keep happening, probably in the types of annual epidemics that one sees in other areas where cholera is epidemic. We do a lot of work with the genetics as well. In this case, looking at the genetics of this particular microorganism. And what's particularly interesting about this is that here we had a single clone dropped into a country, an island. And so it's given us the opportunity to actually watch the evolution of this particular bacteria as it spreads through a population and through the environment. And what we find is that, again, you see it sort of showing all the standard things one expects with rapid evolution as it moves into new areas. And we also see it changing fairly rapidly. This raises questions about things like long-term virulence, environmental survival, vaccine efficacy, because we truly are seeing significant changes as this bacteria essentially sets up housekeeping in a new home. And we still have questions. Is the causative Vibrio cholera microorganism establishing long-term environmental reservoirs in Haiti? Unfortunately, our data says it probably is. How can Haiti best control and limit further cases of cholera within its borders? We're still working on that one. We work closely with the Ministry of Health and with other investigators. There are a variety of plans that have been proposed. But by using mathematical modeling and all the other tools, scientific tools that we have, we're in a unique position to try to figure out the best strategies. The problem is that it may not just be Haiti now. And again, this gets back to the concept of these are pandemic pathogens. Haiti appears to be the source for recent outbreaks of cholera in Cuba and Mexico. For those of you who may have been following the news, I think the case count as of today is 176 cases of cholera in Mexico, down south of Mexico City. The molecular genetics show that this is the Haitian strain that has moved to Mexico. Now, where did it come from? Again, I'm not sure how well you can see it, but there's not that far between Haiti and Cuba. And actually, some of the strains may actually have moved in the environment across that strait between Haiti and Cuba to have gotten into Cuba. We don't know how big the Cuban epidemic was because we don't have data from Cuba. But sort of the under-the-table information we're getting suggests that there was a fairly substantial epidemic in Cuba, probably triggered by the Haitian strain. Now we know it's moving into Mexico. Just within the last several days, we've got additional case counts coming in from Mexico. The Mexican epidemic, which is starting to emerge, is starting to look very concerning. So again, we talk emerging pathogens. We're talking about something that's constantly changing, spreading, and when we're dealing with these pandemic pathogens, such as cholera, an ancient pathogen, been around for a long time, but still very capable of moving rapidly through populations, across continental boundaries, into new countries, as we're seeing today with cholera. And at this point, we're going to shift gears a little bit and move into a discussion format and talk about some additional pathogens. Right.
Okay. So, hi everybody, I'm Maryn McKenna. We have, I think, Roz, we have 30 minutes all told in the session. So we'll talk amongst ourselves with you listening for about 20 minutes, and then we'll open it up to questions for the last 10 minutes or so. So I think, Glenn, I'll start with you. The potential spread of cholera that you just mentioned to Mexico and Cuba is really concerning. And to me, it really enforces the unpredictability and undetectability of pathogens as they move across national borders. As you said, to pathogens, borders are irrelevant. Can you talk about that a little more?
I think, again, for me, from one aspect, this is what makes this such a fascinating field. From a public health standpoint, this is the problem, is that you have pathogens that can be carried by people, can be carried by insects. And every time a 747 lands in Orlando from some place that has vector-borne diseases, at least 10 mosquitoes get off along with the passengers, carrying their own particular set of cargo. We've been involved in a number of studies that have looked at global transport routes. There is no question that today, as you know, it is so easy to get from place to place. The diseases come along with the people. The lice come along and remain relatively friendly. The pathogens may not be so friendly. So we clearly, at least in the Caribbean region, we have major concerns. There is a lot of movement. Again, how it got into Cuba, we're not quite sure, but that's a very small strait across there. We're not quite sure how it got into Mexico, but clearly in Mexico it's there and it's starting to rapidly expand. It raises questions about the entire Caribbean and Latin America.
And what does that say about, if we're talking not just about the movement of large numbers of people, as for instance the UN troops to Haiti, but potentially a single tourist, as happened in 2003 with SARS? What does that say about the problem of detectability in time to stop this movement?
That's the real problem, is that it only takes one. And again, I think cholera in Haiti probably was a single individual that has now caused a massive epidemic in Haiti and may well be causing massive epidemics in this entire region. SARS is the same way, and actually I'm going to bounce this over to Greg. You want to?
Right, so Greg, so when we think, I think most people of the pathogens that we're most concerned about right now, the ones that we think of as having massive pandemic potential, the two that come to mind are MERS coronavirus and potentially the H7N9 avian flu in China. So you were telling me before we got started that maybe uniquely in the United States, your institute has a good working relationship with the Saudi Ministry of Health. So maybe you could tell us a little bit more about what's known about MERS now and what your group is finding out.
Okay. Yeah, so we've been fortunate to work with the Deputy Director of the Ministry of Health in Saudi Arabia, Dr. Ziad Memish, I guess for about 8 years, on various different epidemiological infectious disease problems. And as with most international research, foundations for success in these collaborations are based largely on trust and personal relationships. So having that as a background, we're able to do things I think that other folks are not able. As an example, we have 6 studies in collaboration with the ministry right now. These studies are multidisciplinary in their approach. We're focusing on the zoonotic diseases that, in addition to MERS coronavirus, that might be transmitted with the 14 million people that annually make the Hajj or the Umrah pilgrimages. And the Saudi government actually has stood up as an example for the rest of the world, a center of excellence sanctioned by the World Health Organization in mass movement of people. And they actually are leading in this discipline, mass gatherings as it's called, as they've had so much experience with the in-migration of people and animals for their pilgrimages. So we're learning as well as partnering with them. In fact, just last week, or about a week ago, we had a faculty member and a PhD student helping them with aerosol sampling in association with the movement of the people, looking for various respiratory viruses, as well as mosquito sampling. They're having a huge problem with arboviruses. So Saudi is acutely aware of the MERS coronavirus. We're helping them actually looking at some potential occupations that might be an increased risk, but also they're very interested in other zoonotic and environmental hazards that are associated with the in-migrations of people. So I would say that, in supplement to what Glenn has said, the Emerging Pathogens Institute is a relatively new concept in the United States, and we're very fortunate in the state of Florida to have this institute that combines animal, plant, human, environmental professionals together to look at these complex issues.
So a couple of follow-up questions to that. First, do you have any sense yet out of your work of whether the animal reservoir of MERS might be better defined? There's been talk about camels and talk about bats. What do you think you see?
Well, I'm aware of the literature with respect to potentially several viruses, reservoir that are coronaviruses in bats, and the serologic evidence of dromedary camels having a high prevalence both in Saudi and some other countries, Oman. I think the question that most people are wrestling with is because a lot of the cases don't have direct or even an alleged indirect contact with either camels or these bats. How much of a impact are the animals playing? And maybe they were the original source. There's a strong element of thinking that there's probably a domestic animal amplifying reservoir. In our zoonotic disease literature, you might know that alligators were an amplifying reservoir for West Nile virus here in Florida. Baby alligators, the people around the alligator farms are having a high prevalence of disease. So potentially there's a domestic animal that's playing a role in causing high virus loads, as we say, that somehow are sharing the virus with humans. So the Ministry of Health has engaged international leaders, FAO and OIE are the acronyms for animal studies, and asked which animals would they recommend studies be done upon. And right now, the ministry is about to engage in some large surveys. The difficulty in implicating an animal is that the serologic assays are very imprecise and imperfect. And so having a positive serologic assessment in a camel, we're not quite sure what that means. It could be due to other related viruses. We're just not sure. So there's a lot of work that needs to be done in that area in assessment of the animals. But I think the bat information is pretty solid, even though some of the sequencing published so far has been a small segment of the genome. I think most people would believe that that's a biological, defendable, plausible, if you will, source for the virus.
So we're about 2 weeks out now from the end of the Hajj season. Is there any cross-border spread that's been seen, and how long do we have to wait to know if there is?
Well, I think the window for exposure and incubation is now thought to be about, the outside part of it is 2 weeks, and so we may not see a lot of new cases in association with the Hajj that just occurred, but you need to know that the Hajj is only 2.5 million people, and they're expecting in the neighborhood of 11.5 million people to do the Umrah, which is also a more abbreviated pilgrimage, and they have some 128 countries that send pilgrims, and so even though perhaps the Hajj is not thought to be a catalyst for immediate worldwide distribution of MERS coronavirus today, we could certainly see something different in Umrah. And our Saudi colleagues are acutely aware of this. They are engaged with the WHO in educating people. They are sharing a lot of information quickly with respect to their cases. They've stood up tertiary care centers with the appropriate diagnostics. I know there's been some criticism, but if you look at it from their perspective, you might say that they've been pretty transparent, at least in comparison to what we've seen in the past with major pandemic threats. And so there's an awareness that this could spread and the whole paradigm could shift into an international problem very soon.
So thank you for raising the transparency issue so I didn't have to. Yeah. It sounds, I know you're aware that there has been a lot of concern and criticism of the KSA government of not apparently volunteering details of cases fast enough in the opinion of a lot of the, well, it appears a lot of the rest of the sort of world public health structure. But it certainly sounds as though you have a different point of view about that. So is there anything out of your knowledge that you can share with us about what's going on behind the scenes that might make us view that differently?
Yeah, let me just share with this audience in particular. It's really easy to beat up on people from different cultures and to listen to one side of an argument, and harder to engage, particularly if there's a language barrier, people in other cultures. Having this personal relationship with the leader in the ministry, I happen to know some of the background for some of the friction. A lot of it has to do with litigation. and lawyers. You might know that the Saudi Ministry of Health has been very much embarrassed and even deceived in its partnership with other academic and research institutions. There's a virus called the Al-Qurma virus that was taken out of their domain and published, and basically they were embarrassed. And then recently the Erasmus group sought to patent the MERS coronavirus. And so putting intellectual property ownership on viruses or working without these international partners can be a tremendous political, economic, and otherwise offense. And so what the Saudi government did is they engaged Western lawyer group to protect their interest. And the Western lawyer group is requiring international partners to sign agreements that such that that the Saudi government can control somewhat some of the intellectual property issues, and the partners will not make money. So just know that that's a real fly in the ointment right now, and a lot of it is being driven by those of us in the West who are seeking either economic gain by patenting a naturally occurring pathogen and then deriving diagnostic revenue and vaccine revenue from that patenting, which I think is a real thing to focus on as a science writer. Is this ethical? If a new element was discovered in our environment, could you patent that element? You know, and it's similar to what's happening now with these viruses. And so because of this, the institutions and the government institutions in particular are reticent to sign these agreements because they want their autonomy and their freedom. And so what is happening, they're bashing the ministry. When, from one perspective, these institutions have been a catalyst for this new barrier because of past behavior.
So I want to turn to the other disease that I think everyone thinks of as a potential pandemic, and that's flu. I know, Greg, you just came back from China, and Glenn, you've been concerned about pandemic flu for years. Is there anything you can say about H7N9 first, the avian flu that's now circulating in China?
Yeah, I think number 2 on the hit parade as far as emerging threats behind the MERS corona that has me concerned is the H7N9. And we're fortunate, it looks like, that the measures that the Chinese government, particularly the closing of the open bird markets in areas with a high likely poultry endemicity, has somewhat reduced the infections in humans. But there is a lot of concern. Now, what's interesting, I think, in China is that they have the infrastructure already in place to do major interventions. And if, for instance, if they want to close the poultry markets, they close them. And so it's very interesting to see the engagement of public health professionals like ourselves influencing the government and the rapidity with which they can affect a change. I can say that the scientific resources, the infrastructure, the sequencing, the information that's being collected with respect to H7N9 in China is unprecedented. It's really amazing, the facilities they have and how quickly they have brought up new diagnostics, even vaccines, and are supplying the world with reagents right now in combating this new threat. So I'm very impressed with what's going on in China. And moreover, they're beginning to see the need to embrace an international, interdisciplinary approach that we embrace here at the University of Florida. Glenn mentioned it earlier, the One Health approach, because we recognize that H7N9 and other viruses, such as swine influenza viruses, are multidimensional in their prevalence. You can't just be a veterinarian and expect to control it, or on the human side. You actually need to partner with people in the environment, the veterinary, and the human side to solve these problems. So much of my last month was spent engaging these very senior institutions and encouraging them to adopt a One Health approach to these complicated problems. And we're seeing a lot of inertia in that area. To have people come out of their various institutions and partner in designing studies and interventions across institutions.
So it's interesting to me that we're so concerned about a flu that since about the spring has produced about 150 cases in China, where we have a really interesting flu story emerging here in the United States that's much less well documented, and Glenn, you referenced it. So can we talk for a couple of minutes about the H3N2 flu, which I think for those who are flu geeks, this is the state fair flu that's perked along for the past couple of years.
Yeah, that's a plant perhaps. This is my passion, pigs and influenza virus. You know, we're so much like pigs in our biology that in contrast to other interspecies transmission, such as avian flu and equine horse flu and marine mammal flu, we don't see the cross-species occurrence, but we really do with pigs and people. And what is happening is we have introduced our flu viruses into pigs. And because the current way we produce meat in pigs is so vast, there's a constant group of animals which are naive and can continue to replicate the virus. And so we're seeing the pig farms, despite their biosecurity, as a source for novel virus generation. And Maryn has mentioned the 2009 pandemic. Of course, we think very strongly that the pig production facilities, the modern ones anyway, have played a role in at least the spreading of that. Well, now a new progeny virus has come from the 2009 H1N1, actually several, but the one that's captured the most attention, and there's a new report in the journal Clinical Infectious Diseases, is called the H3N2 variant. And that virus is like the H7N9 virus. Humans are serving as sentinels. In other words, in China, we don't know where the H7N9 virus is geographically until we see a human case. Then we go find it in the chickens. Well, that's happening here in this country with respect to the H3N2 variant. And so we know that people are being infected when they seek medical care and they have severe disease. There's probably a lot of mild disease going on. And then we find it in the pigs. The problem is in modern pig production, there's no motivation for them to study this virus. It doesn't really make the pigs sick, just like H7N9 doesn't make the chickens sick. And there's not a good way. We have a few studies in collaboration with US CDC and the USDA to get a mild estimate of the problem, but we don't have the motivation, the mechanism, the funding to really get a good handle on how fast it's spread. So what we thought was very much a contained problem in a report that's just coming out has now affected over 300 people in the United States in 10 states. So it's spreading. We have no good pulse on it. We don't have any good interventions in mind. The US CDC has a vaccine construct that they've prepared, but you know, it would be nice to have an understanding of how this is being amplified and what are the risk factors for cross-species infection in association with pig farming. But frankly, that's not being done. So we think we can do that in China because China government owns a lot of pig farms, and they have an even bigger problem with more viruses than we do and larger farms. And so the Emerging Pathogens Institute is heavily engaged in studies of pig zoonoses in China with our partners, where we think we'll have some more interesting work. But that's something to keep your pulse on as a science writer. You know, today's complex systems of food production, meat production, we have to come out of our parochial ways and do interdisciplinary research. And frankly, there are a lot of barriers to doing that today.
And
so
I would just add very briefly, this gets back to way back at the beginning of some of the first slides I showed. The idea that what you see are viruses able to move now into new niches, in part because of the way we're changing things. And so things like pig production. Used to be you had a couple of pigs in the backyard. Now you raise very large numbers. How many in a standard pig farm?
Yeah, some of these farms are 120,000 head, which is huge. Yeah.
And so what it does is, because of the way we are changing the way we manage things, we are opening doors. You've got 120,000 pigs in a single farm. They can spread all sorts of interesting viruses among themselves, amplify those viruses, and then those viruses can jump into humans.
So we have an uncontained, under-surveilled, cross-species epidemic occurring in this country involving industrial food production.
Correct.
That makes me so happy. I'm sorry to be a ghoul, but really. Okay, that's our 20 minutes. I've had my fun. There are microphones. If anyone has questions, I can certainly keep asking questions if you don't, but I bet you do. Let's start. We'll go from side to side. Tom, let's start with you.
Hi, Tom Ulrich, Boston Children's Hospital. One thing that neither one of you addressed, but which I think could be of interest to the room, is the topic of climate change and the spread of infectious and communicable disease. I'm from Boston. The worst thing we really have to worry about these days, which is still pretty bad, is West Nile or Eastern equine encephalitis, at least in terms of mosquito-borne disease. Is there going to come a point when my kids have to be worrying about malaria or dengue or things like that showing up, you know, when they go to the ball games at Fenway Park? And go Sox.
You know, my favorite pathogen, cholera, actually is very sensitive to climate change. As I said, what triggers its bloom and spread into human populations are increases in temperature. And so cholera is clearly increasing at a global level, and there is a great deal of concern that it and other Vibrio species may well be being driven by climate change. The problem is right now the changes we're seeing are relatively small. It just happens that for cholera, those changes are right at the point between poor growth and very good growth. Mosquitoes are a little trickier. Right at the moment, we're not seeing clear evidence of impact on climate change on spread of malaria and other vector-borne diseases. That may well change. Clearly, this is an extremely important area that we need to keep a very close eye on.
Bob, let's go to you.
So I'm wondering, how can we as journalists help or hurt the spread of pandemic disease, assuming that we're already not letting our kids kiss pigs? So.
I think part of it is an awareness of what's going on, a recognition of the potential for international spread, a recognition that public health is important. As probably most of you are aware, public health departments, particularly here in the United States, you know, they've gone after them with a meat axe in most states, including Florida. And so we've got a good public health system, have had a good public health system, but it's rapidly deteriorating in this country. And unfortunately, the thing about public health is if you are succeeding, nobody sees what you're doing because nothing's happening. And this is an instance where I think there needs to be an increasing awareness that we do live in an age of pandemics. And to deal with that, we've got to have, you know, our best game, you know, our A-team in place in public health. That's not happening.
This side, please.
You sort of anticipated, maybe if this is a segue, during the recent government shutdown, one of the concerns voiced by our infectious disease people was the fact that the CDC could not track flu in this country for roughly 2 weeks. Fortunately, things got back together after about that time. If that had extended, could you comment on what kind of risk that would be to public health in the country, especially in regard to flu?
Well, I mean, it'd be a tremendous risk. Our CDC, I think, is viewed worldwide as the finest, if not one of the finest in the world, and in that it combines both classical public health with research, and to lose that would be a major blow to the United States and to the world. The leadership, as a good example, they have engaged for now maybe 10 years the development of national centers for influenza in many countries and brought modern diagnostic to places that were sometimes without electricity. And so I have the highest respect and praise for the CDC, and to lose that would be just a terrible loss. And so you as science writers can find your champions in public health and do what you can to protect them. Many people would argue vaccines are another thing to focus on, and they're under attack with various different misunderstandings of their value. But I think it would be a huge blow to lose the CDC for any length of time.
I would chip in there, though, and say as well, I think the CDC is the high visibility arm. What concerns me as much are the state health departments. Yeah. It's at the states that we are having major blows to public health in a number of states, where they're basically just dismantling the public health departments. And so I think that's a major concern, because it's really, the CDC deals with it at a national level, but the guys on the ground are the state and the county and the city health departments. And that's where we get the feedback that things are really hurting.
So I just want to flag that we have 3 more minutes. I know there are more people who want to ask questions than will fit in those 3 minutes. So since we have to be, I believe, out of the room at 10:30, when we get to 10:30, we will officially thank our guests, and then we'll continue the conversation out in the hallway or up here at the stage wherever's allowed. So please go ahead.
Okay. Briefly, 2 things. First of all, this intellectual property issue is not new. For example, one of the major chemotherapy drugs was based on a Madagascar plant. So I'm wondering if other countries are up in arms and doing something legally about this issue. The other issue is, if I remember correctly, the cholera vaccine is not very effective, and I'm wondering if there is any work going on to get a better, more durable vaccine.
This gets back, again, unfortunately, to an economic level, which is that cholera vaccine doesn't make a drug company money.
Right.
And the people in poor countries who need cholera vaccine can't pay for it, or they can't pay the prices that the drug company needs to be able to get it through US approvals. And so what's happened is that cholera vaccine, actually the only current commercial source of cholera vaccine, is a small drug company in India, because they're the only ones who'll make it. And this gets into a much larger question about the way in which we develop drugs, and in particular the development of drugs like cholera vaccine. I mean, at a personal level, we developed a dynamite cholera vaccine, which was patented. We actually tried to move it forward, and we were told point blank by all the big drug companies, we are not in the least bit interested in cholera vaccine. It will not make us any money.
What about the intellectual property?
I think there's a major understanding that intellectual property barriers are harming science today. I'm not quite sure how we solve that, nor am I sure of the appropriate forum. But even the Margaret Chan, the WHO director, has admonished those of us in science about putting these barriers forward, particularly with respect to a pandemic threat. So it's a developing problem that is growing, and it's going to have to be dealt with.
On this side, thanks very much.
Hi, I just wanted to ask about the cholera coming back to Haiti after 100 years, especially with how it can be in environmental reservoirs. I just need to know what happened 100 years ago and how was it eliminated then?
Well, we're not sure. The 100 years is sort of a number you pull out of the air. The fact is, there really never was any documented, within the modern era, there has not been documented cholera in Haiti. You know, it might have been back in the back, you know, there were cholera epidemics in the United States 150 years ago. Could there have been cholera epidemics in Haiti? Maybe. We don't have the history. So you hedge and you say, well, for at least 100 years, the island hasn't had cholera. The problem now is the island clearly has cholera, and you get into this whole issue of environmental reservoirs, and that's what's really scary.
So do we have a sense of what happened 100 years ago to make it so that there wasn't cholera anymore, or is that?
Well, there may have been cholera there, but we just don't know. So within modern scientific history, we have not documented it in Haiti until 2010.
So we are out of our official time. Please thank our guests. I know they'll keep talking.